Welcome everybody to the Brain and Behavior Research Foundation monthly Meet the Scientist webinar series. I'm Dr. Jeff Borenstein, the President and CEO of the Foundation, and your host and moderator for today's webinar. The webinar is entitled Research Discoveries in Schizophrenia and is presented by Dr. Daniel Weinberger of the Lieber Institute for Brain Development. The Brain and Behavior Research Foundation is dedicated to funding research around the world to identify the causes, improve treatments, and ultimately develop cures for mental illness. The Foundation has awarded approximately $300 million in NARSAD research grants over 25 years. I'm delighted to introduce Dr. Daniel Weinberger. Dr. Weinberger is the Chief Executive Officer and Director of the Lieber Institute for Brain Development. He came to the Institute after leaving the National Institute of Mental Health. Dr. Weinberger has been supported by 2 NARSAD Distinguished Investigator grants, has received the Foundation's Lieber Prize for Outstanding Achievement in Schizophrenia Research, and serves as an active member of the Foundation's Scientific Council. He is also a professor of psychiatry, neurology, neuroscience, and human genetics at the Johns Hopkins University Medical School. Before we begin, I want to let you know that this is an interactive event. We'll begin with Dr. Weinberger's presentation, followed by a question and answer period. To submit your questions, please use the Questions tab of the webinar control panel. You can submit your questions throughout the presentation. I will present your questions to Dr. Weinberger, and he will address as many of them as possible. And now, I'm pleased to present my colleague, Dr. Dan Weinberger. Dan, the floor is yours.
Thank you very much, Jeff. It's really a great pleasure for me to be with you all. I can't say strongly enough how important the Brain Behavior Research Foundation is to the landscape of research about mental illness. It's played a very big part in my work over the past 30 years, and I'm just delighted to be here to share with you some of the late-breaking work that's going on in our efforts to understand schizophrenia, but other really developmental brain disorders that impact on human cognition and emotion. Let me start off by saying that we have learned in the last decade more about the causes and nature of mental illness than we have known in all of past history. This has been an extraordinary decade for scientific insight and discovery. And there's no question that the royal road to reducing stigma and to improving the lives of affected individuals is through scientific research and discovery. The pace of this discovery effort is quickening very, very dramatically. So I wanted today, sort of in a sort of whirlwind tour, give you a feeling for some of the very late-breaking science and where the current thinking about the causes of mental illness are, and how some of this thinking will lead to new therapies. So as I move through these slides, this is where I'm going with this talk. We're going to give you some insights into late-breaking discoveries and then hopefully provide some ideas about how this will lead to improving the lives of sick people. Let me start off by reminding you where we were at the turn of the 21st century. We really had a great deal of information about what illnesses like schizophrenia looked like, what they sounded like, what they seemed like. To a very great degree, we even knew something of what they felt like, but we actually knew absolutely nothing about what they actually were or are at a basic scientific level. This is about what we knew at the turn of the 21st century. Diagnosis is a subjective process based on non-specific phenomena. We knew from family and twin and adoption studies that most of the variance in risk across the general human population was accounted for by genes. But this was an abstract concept that had no hard, concrete reality or biology linked to it. We knew that environmental adversity in early development, particularly during intrauterine life or around birth, slightly increased the risk that someone would manifest schizophrenia or related disorders in their lifetime. There was evidence of subtle deviations in childhood development. Many studies looking at cognition and these brain imaging studies that really gained a lot of momentum starting in the 1980s suggested that there was subtle functional abnormalities of parts of the brain called the frontal lobes and the temporal lobes. Over a century of studies looking at brain tissue of people who died with these disorders had not discovered anything very dramatic, certainly nothing diagnostic. There was some very subtle nonspecific abnormalities found. And we also knew that antidopaminergic drugs, for whatever reason, were the therapeutic agents of choice at this time. Let me just review a few of these insights. It was pretty clear from a number of large population-based studies that signs of brain developmental problems were present from very early in life. This was a study from Finland, one of a number of studies that showed that if you look even at very early classic developmental milestones, such as the age at which an infant stood up, which is generally about 9 months. That's the average age at one stands up. On the x-axis here, where it says age, is the prevalence of schizophrenia at different ages of life. On the y-axis, where it says cumulative incidence, it says what is the likelihood that out of 100 people in the population, there's schizophrenia in the population. So 1 is about what you get if you stand up at around 10 months. 0.6% of the population is 9 months. If you stand up 3 months later than the average mean for the population, you had a 2-fold increase, 2 to 3-fold increase likelihood that you would have schizophrenia as an adult. Now, this only increased your risk to about 1.8%. So most people who stood up at 12 months did not develop schizophrenia. This did not increase your risk to such a degree that you would likely have schizophrenia. But what it demonstrated was there was something about the people who 25 years later or 30 years later would manifest schizophrenia that could be found in evidence of early developmental milestones, very early in life, suggesting there was something about how the brain was being put together that wasn't quite what the average would be for the population. There have been many other studies of this. This was a study that Tom Hyde did now at the Lieber Institute when we were at the NIH, looking at other very well characterized developmental milestone, which is when you gain control of urination, and this is so-called enuresis. And patients with schizophrenia have about a 3-fold greater likelihood of having had child enuresis than the general population. Interestingly enough, their healthy siblings who were not ill psychiatrically had about a 2.5-fold increase in the likelihood of their having enuresis, suggesting that not only was there something about brain development that could be observed at this kind of characteristic, enuresis, and urinary control, which by the time children get to be 5 or 10 is perfectly normal, even in people who ultimately develop schizophrenia. But this suggests that there was some difficulty in getting the programs of brain development organized quite the way they should be, even with respect to this, what's classically called a neurological milestone. So there were many other factors that have been linked to an increased frequency that somebody would manifest schizophrenia in a lifetime. Some of these are illustrated here in this slide, whether you grew up in a city, whether you had another psychiatric disorder, an older father, social problems. All of these things had been linked weakly to an increased likelihood of schizophrenia, suggesting that there was something about the factors of brain development that at this early stage of life didn't manifest as schizophrenia, but they manifested as other subtle problems. None by themselves would cause a parent to take a child to a pediatrician or the pediatrician say this is abnormal, but in aggregate they suggest that all the programs of getting the brain wired quite right were not working completely appropriately. The only one of these things that was something other than an association was the evidence of genetic predisposition, because genetic predisposition by definition is something about causation. It's not necessarily the result of something else. Genetics is the cause of something else. It can't be the result of something else because we start with our genomes. The first cells are just our genomes. It's a little bit like saying that if life is a monopoly game, genes will never determine how many railroads you own or whether you have monopoly on Park Place and Boardwalk. But genes are the only square on the board that you ever know absolutely objectively. Genes are the go square. They represent the toolbox that you have, the resources, the biological resources that you have to play the game of life with. So it's important to remember when you think about genes for mental illness, these are like genes for common medical disorders. They're like genes for heart disease, they're like genes for stroke, they're like genes for asthma, obesity, diabetes, and even cancer. They're not genes for your fate. They're genes for the risk that you have as you accumulate other lifelong risk factors to manifest these conditions. And what's characteristic of all these genetic disorders, which are called complex genetic disorders, as illustrated in this slide, they're characterized by two things. They're polygenic, multifactorial, meaning no one gene or one factor all by themselves cause anything related to illness. And the other is that they're heterogeneous. Everybody with the same clinical disorder does not have the same factors that contributed to their having it. So for example, in this slide, if you think of everybody with the clinical disorder of interest, perhaps schizophrenia, autism, or bipolar disorder, as being one of these brown circles, you can see that every brown circle has four pieces that are meant to represent the causative factors to getting to have a brown circle rather than a white circle. The fact that there are four means this is multifactorial or polygenic. But every individual who is a brown circle does not have the same four puzzle pieces, meaning that there are different ways you can get to the same state. The other way of thinking about this, if you have heart disease, is that cholesterol, blood pressure, diabetes, overweight are different factors. None of them by themselves guarantee you heart disease. Different combinations in different people can produce the same end result, which is called a heart attack. But how you get there varies across different people, and the different factors conspire in different ways to have a similar endpoint, here mental illness, there heart disease. Now, the notion of genetics as a virtual or abstract concept has changed profoundly in the last 10 years with the availability of the human genome sequence. And this has made it possible for scientists to study genes as real biological entities and not just as these abstract ideas related to inheritance. And one of the very popular strategies to do this now is based on the technological developments that have led to the ability to, in very quick fashion and at remarkably low cost, survey common variants in the whole genetic sequence of human beings. And this is done with these technologically remarkable things called SNP chips. And it's really a chip that's for the most part developed with molecular biological techniques. That is a microchip. It's very tiny, and it's maybe an inch square. And with this chip, you can survey millions of variations in the DNA code comparing one individual to another, and is illustrated here with patient 1, patient 2, patient 3, patient 4. You see a row here which is the genetic code with all these letters that make up the alphabet of the genome, that each individual has some variations in the alphabet. This is why we don't all look the same. This is why we're not all the same height. We don't have the same emotional responses to things. We're different. And we're different because we have different parents, we inherit different genes, and we also inherit different variations across the genome. And when you do these chip analyses, you can survey the entire genome of people and find out where they're different. And you can group a whole group of people with one diagnosis like schizophrenia and compare those differences across the whole genome to a group of individuals who we collect as controls who don't have schizophrenia. And where you find differences that are consistent across individuals with one diagnosis, schizophrenia, that aren't found in normals, you say those differences are associated with the illness state. That's the principle of genetic association. And this is being done now with increasing results. This was one of the first studies called the Psychiatric Genomics Consortium study. It was published in the journal Nature Genetics about 2 years ago. It was a huge sample of people from 30 different populations around the world and 51,000 subjects. And these studies require a very, very rigorous statistics because you're comparing variations across the genome in millions of places. So you have to control for the fact that many of the findings may be chance findings. Because if if you flip a coin many, many times, sometimes it's going to come up heads, sometimes it's going to come up tails, and and there's a lot of chance in which one comes up which. If you have a loaded coin, it's going to come up heads more often than tails, but it's not going to come up heads every single time. So you have to control for the times it didn't come up heads that were not about the loading, that was about the chance. So what was found in the first study of 51,000 subjects was 5 genes that were significant enough that they weren't by chance. And this was very encouraging, but there's one problem in these studies, and that is illustrated by the column here called OR. This is the odds ratio. And while these are clearly genes linked to the risk for schizophrenia, they don't contribute by themselves very much in generating illness in a given individual. What the OR column, or odds ratio, shows you is that if the normal risk for schizophrenia in the general population is 1, meaning 1 person out of 100 will likely have this diagnosis in their lifetime. If you inherit for this gene, for example, MIR137, and it really doesn't matter what these genes are named because nobody really knew anything about these genes till they were found through these research strategies. But if you inherit the form of this gene that is associated with schizophrenia, it increases the probability that you will manifest the illness from 1% probability to 1.1% probability. This is not a very big increase in risk. This is not a finding that could by itself do anything to identify people at risk. But the value of these genes, I'll illustrate in a few minutes, they provide us with clues to causative mechanisms that are biological that we don't have without this evidence. Now, this was 2 years ago. This is the very latest and greatest. These studies keep growing. We keep getting new samples added to these data sets. And the more you increase the size of the samples, the more you find areas of the human genome where genes associated with the causes of these illnesses are going to be found. In the current analysis, which includes 70,000 subjects, 43 samples of populations around the world, there are now over 70 what are called loci, and loci just means regions on the 22 chromosomes, non-sex chromosomes. If you remember, the human genome has 23 pairs of chromosomes. The 23rd pair, not shown here, is the XY sex chromosomes, and the other 22 pairs of chromosomes are called the non-sex or autosomes. There are over 70 regions that are significant enough that they are likely to be real findings related to the causes of schizophrenia. And the other thing you can see is they're all over the genome. There's really almost no chromosome. Maybe the 9th chromosome, for whatever reason, has escaped being included in this list of places in the human genome where across these heterogeneous populations, risk factors have been found. And probably as a sample size goes to 80,000 or 90,000 or 100,000, one of these guys on the 9th chromosome will creep over the high bar and also be what's called significant locus. This is very important because it demonstrates there is no one spot in the human genome that has a monopoly over where something can go wrong that will contribute to the risk for schizophrenia. There are obviously many ways that someone can arrive at this condition based on their genomes. Now, there was a very interesting paper that appeared in the British medical journal Lancet 2 weeks ago that was commented on in the New York Times, the Boston Globe, many sites around the world. Because this paper put together the genomes of 5 psychiatric diagnoses: schizophrenia, bipolar disorder, major depression, autism, and attention deficit hyperactivity disorder. And the paper took the strategy that we know that psychiatric diagnoses are imprecise. We know that there's a lot of overlap at the clinical level across the lifespan of people. There'll be attention deficit hyperactivity disorder as children, then they'll be depressed, and they might get a diagnosis of schizophrenia, etc. So they put together all these conditions, and they did the same study looking across the genome. Were there any genes that were now found in this amalgamated sample of psychiatric illnesses compared to normals? And they found 4 of them. This was a sample of 61,000 subjects, and they interpreted these data to suggest that there were common factors across psychiatric diagnoses. There were 4 found in this sample. It was very hard to know which were the genes, but it was striking that 2 genes showed up that had previously been found in samples of patients with bipolar disorder and samples of patients with schizophrenia. And both of these genes were in proteins called calcium channels. And calcium channels are very interesting because calcium channels are important in cells, in neurons communicating with each other. Calcium channels are critical in the function of many enzymes in cells and in the way cells excrete neurotransmitters. And we've been interested in neurotransmitters for a very long time in psychiatry because most of the available drugs that we use target neurotransmitters. So this is a very interesting finding. There's another perspective on this study that was not mentioned in the study that I want to share with you because I think this study principally celebrated the idea that there were some common variables across psychiatric diagnoses. But there is another side to this study, which is that one also could make note of the fact that there are clearly distinctions between these diagnoses. So if you take a look at the CAC-NA1C finding, which was significant by the standards used in these things, this is the odds ratio across these 5 diagnoses. It increased the probability that you would be in the class of 5 diagnoses rather than being in the normal group. A tiny, tiny amount. If the chances that you'd be in the normal group were 1 out of 100, now your chances were 1.07 out of 100. Not a huge increase. What about if we looked at this gene as it was first discovered in a study only of bipolar disorder, where it was first discovered? And this was a report by Pam Sklar 2 years ago in Nature Genetics. In fact, in a sample of 1/3 the size, where they only looked at bipolar disorder, they actually had a stronger effect. So one could also interpret these data to suggest that while there are common factors across psychiatric diagnoses, there really are also going to be clearly distinct factors, both genetic and otherwise, across these diagnoses. So let me start with a question here, because I've shown you some evidence about how genes are being discovered, and there are going to be many, many genes of interest. But these associations are weak. At the individual level, any one of these associations do not increase risk very much. Why is this? So there are a number of answers to this, and I'm not going to dwell on most of these. I've mentioned to you heterogeneity. Heterogeneity means that every person with this diagnosis does not have the same illness. And so to expect a given factor to be meaningful to everybody with a diagnosis that is a subjective label that people get is probably naive to begin with. We know that the environment's very important. I'll talk about this. I'm not going to talk about much about the possibility that some people may be ill because of very rare things. I'll mention it briefly. Epigenetics is an increasingly interesting area of genetic science where factors that affect the genome are not inherited in the sequence, but I'm not going to talk about that. I'm also not going to talk about epistasis, which is the idea that no gene by itself really can explain much unless you look at it in the context of other genes. But I do want to mention the idea that genes don't encode for psychiatric syndromes. And all of these studies are based on looking at how a gene translates into one of our arbitrarily grouped syndromes. So these are the 3 take-home messages from the last few minutes of this talk. The genes for psychiatric disorders are not for psychiatric disorders. Genes for risk is critically dependent on context. We have to understand an individual's genetic background and their environmental background to understand how any given gene translates into their particular illness. And then finally, genes will impact on outcome and treatment response and lead to new therapies. Genes are mechanisms of illness. Genes that have been discovered are pathways to understanding brain development and brain function. We now have the first clues to the causes of mental illness and to what, at the basic cellular and brain level, these conditions are. All the treatments that we use in psychiatry today, none of them were based on any fundamental understanding of the causes or pathogenesis of mental illness. Now that we have clues to these causes, we have to do better than we've done in the past in the strategies we've had to find drugs. So when I say genes don't encode for psychiatric syndromes, of course genes are only about cells. And when you inherit a variation in a gene, it creates some bottleneck in the biology of a cell. Cells make brains, and our psychiatric syndromes are the emergent phenomenon of a brain that has been wired based on a molecular glitch because of the inheritance of genes in a cell. This is very analogous to thinking about how the musical performance of an orchestra is disturbed at the level of a few notes in the score of so the genes are the score that the cells, who are the individual instrumentalists, are reading. There are woodwinds and brass and strings that are distributed neural systems of the orchestra. What we hear is the music. And there are obviously many steps along the way to produce the symphony that we hear as music. And there are, as you well know, there are many, many little glitches that can occur in the music at multiple stages that can be buffered. They can be buffered by the individual instrumentalists. They can be buffered by the systems in the orchestra. And we don't, at the gross level of our auditory systems, necessarily hear the music as having abnormal sound. But yet, at some point, the buffering can be abnormal. The assumption here that genes are not about these psychoses, but about how a cell develops and grows and operates, and ultimately how a cell works, means that we can study people who don't have psychiatric illness at the level of how their brain works or at the level of cell biology if they're related to people who manifest psychiatric illness, because they also will inherit some of these genes. And the way you do this is by studying healthy people who have inherited risk genes but don't manifest illness. And the early studies that were done, this is work by Terry Goldberg when he was with us at the NIMH over 20 years ago. The early work on this was to study healthy twins of patients with schizophrenia. Twins have all the genetics of mental illness, but they don't have something else that translates into illness. And when we studied healthy twins, the unaffected twins of patients with schizophrenia, on some of the characteristics that were associated with illness, but not the diagnosis, such as cognitive problems, we found that many of the healthy twins, even though they weren't ill, had some of these cognitive problems. One can go one step closer to how the brain works and don't study any manifest behavior at all, but study brain processing of information using novel brain imaging techniques. This, even if you study healthy siblings of patients with schizophrenia, unlike twins who share all their genes, siblings share 50% of their genes. And when you do this, you find similar patterns of brain activity, abnormal patterns of brain activity, even in patients even in subjects who are perfectly normal, but share 50% of the genomes with individuals who are ill. This suggests that the genes for mental illness are about how the brain works. They're about how the brain develops and works, even in individuals who have other factors, such as not enough genes, or environmental protective factors, or perhaps genetic protective factors that result in those risk genes not penetrating all the way to the level of clinical illness. But you can study the effects of these genes at other levels of biological function, here brain processing of a working memory task, which we know is abnormal in patients with schizophrenia. This suggests that if we take genes linked to psychiatric illness from other areas of science and look at them in the context of how the brain works, we should see very strong effects of those genes. So here's a study by Christy Bigos, when she was also at the NIMH, now at the Lieber Institute for Brain Development, looking at this most popular gene of all the genes that have come out of these studies, the calcium channel gene. And here's the calcium channel gene, and we're looking in normal human beings now. These are not siblings, these are normal people, but now we're defining people by the risk-associated variation in this gene. And this is the so-called, these are the letters of the genomic alphabet associated with the risk for schizophrenia. These are normal individuals, and we're asking whether these normal individuals are more likely to show this pattern of function of the prefrontal cortex that's analogous to schizophrenia than are normal individuals with these other alphabets in this gene, and they are. And this is a hugely significant effect. If you remember, I showed you in the study that found this in the original sample of 10,000 bipolar subjects, the p-value here, which is the level of significance, was 10 to the minus 8. Here, it's 10 to the minus 109, because this is a much stronger effect that this gene has than it has in our psychiatric diagnoses. I mentioned to you genetic risk is critically dependent on context, and everybody has a different genome than everybody else. We are all unique at the level of our genomes. And so any gene that shows risk at the population level will have individual effects at the individual level. But it's not just genes that determine the effects of other genes. The environment determines the effects of other genes. Genes interact with the environment, and this interaction can modify the expression of the individual effects of genes. The environment can exaggerate effects, it can compensate for effects, or it can lead to novel effects. Let me illustrate one example of this. We've known for about 100 years now that the histories of patients with schizophrenia as a population, not everybody, but as a population, have increased likelihood of obstetric complications. And there have been many studies of this. This was a very nice study from New Zealand showing a clear increase in risk. This was a prospective study done where they followed people for 30 years from birth, that having obstetric complications does increase risk of schizophrenia. We looked at this in terms of genes, and all I want to show you here is here's some genes, these are family studies, here's the likelihood, again with this odds ratio, that someone across about 400 families would manifest schizophrenia if they inherited different forms of these genes. And here's the odds ratio if they have no history of obstetric complications. Here's the odds ratio if they have the gene plus a history of obstetric complications. And now you can see in this study that these effects become quite serious when you also have the gene of interest and the obstetric complication. So the odds ratio of the gene alone without the obstetric complication is not noteworthy, but it goes from 1% to 7%, 1% to 4%, 1% to 12%, 1% to 9%. The genes in the environment interact in a non-additive way. The last point I want to make in this talk, well, the second to the last point, is that there are rare cases of patients with schizophrenia, just like there are rare cases of autism, probably more common in autism than schizophrenia, and in ADHD, and in bipolar disorder, and these were originally found most frequently in intellectual disability, where there are glitches in the genome, not just at the individual letters in the alphabet, but whole regions of chromosomes get fouled up during cell duplication, cell division, when chromosomes make mistakes when they replicate themselves. And these mistakes can be that stretches of a chromosome are deleted, stretches of a chromosome are created, some are duplicated, etc. And we've known for a number of years that the most common deletion in the human genome, which occurs in about 1 out of every 3,000 births, results in the deletion of about 30 genes on the 22nd chromosome. And this causes a syndrome called Velo-Cardio-Facial syndrome, which is a syndrome associated with mild intellectual disability, some facial configurational abnormalities, cardiac problems. But about a third of these people develop psychosis when they reach early adulthood. About 40% of them have ADHD as children. Another 30 to 40% have autism spectrum disorder. And so this is a deletion that about 1 out of every 200 patients with the diagnosis of schizophrenia has this. And in those cases, this is probably the cause of that syndrome. We know now with the technologies to scan the genome that this is not the only rare so-called deletion or copy number variation that's found with increased frequency in patients with schizophrenia. And overall, there's other parts of the genome which is just illustrated in this slide. Some are on the 16th chromosome, some are on the 15th chromosome, some are on the 1st chromosome, some are on the 2nd chromosome, just like those loci I showed you. They're all over the genome. These are much less frequent, but overall, somewhere between 2 to 5% of patients with the diagnosis of schizophrenia have one of these rare so-called variations in blocks of DNA. How much they contribute to individual risk is still uncertain, but they contribute more than those GWAS hits that I showed you. I want to make the last point. I'll try to finish here in 2 minutes. Genes do impact on outcome and treatment response, and they will lead to therapies. And the reason they do this is because genes are about 2 things. They're about fixed traits that you inherit from your parents, like the color of your eyes or your hair, but they're also about the day-to-day biology of cells. We don't expect to change people's genes, but we expect to be able to change the effects that those genes have in cells. Every one of these things here is a protein. Every one of these proteins is encoded by a gene. A number of these proteins are proteins that have been associated with risk for schizophrenia. By understanding how these proteins work in a cell, we can identify targets for improved treatment. Here's one illustration of this. We've been very interested in understanding how cells fire electrical impulses. Cells fire electrical impulses by changing their activity and doing what's called an action potential. These action potentials are critically determined by the inflow and outflow of ions across the cell membrane. And the critical ions are sodium and potassium. One of the genes that's been associated with schizophrenia is a potassium channel called KCNH2. This potassium channel is the critical target of certain drugs like clozapine. And it's been shown now, and this is just a paper that made this point, that variations in this gene that are associated with schizophrenia rather strongly predict response to available antipsychotic drugs. And in fact, in the CATIE trial, which was this big trial across multiple populations in the United States, individuals who carried the form of this gene that was linked to risk for schizophrenia were 5 times more likely to have finished the CATIE trial than individuals that didn't. And this is a new target for the development of antipsychotic drugs. This is a strategy for turning genes into molecular mechanisms of association. We now have a roadmap for doing this. I'm not going to go into this in any detail, but we have a roadmap for finding genes from a variety of clinical studies, translating these genes through an exploration of how these genes affect the development and function of a human brain into molecular mechanisms of genetic risk. These molecular mechanisms are used to build cell models, and there are very sophisticated strategies now based on the development of so-called stem cells from human beings, stem cells that can come from even adult human cells. This is now the basis for building models, both cell models and animal models, which can be used to find new therapies. And this is the strategy that I think is going to be increasingly used both in private institutions. This is our roadmap at the Lieber Institute, and it's also the roadmap being used in industry now. So I've tried to give you a whirlwind tour of where I think some of the late-breaking science is and some of how basic science will lead to real strategies for improving the lives of affected individuals. These are my conclusions. Most complex behaviors are the result of multiple factors that interact biologically. Genes are the first objective clues to the causative mechanisms of psychiatric disorders. There are many developmental pathways to what we call schizophrenia. Genes for schizophrenia likely have their effects on liability or risk, by influencing brain development, the genetics of psychiatric illness is the game changer, both in understanding mechanisms and in finding therapeutic targets based on causation, not phenomenology. I thank you very much for your attention. I'm very eager to answer your questions and hear your comments.
Dan, thank you very much for an extraordinary presentation about a very complicated issue, but presented in a way that the lay public can understand. And this is exciting. And based upon a number of the questions that we've received, really people realize how important these findings are and the potential. One of the areas that we've received a number of queries about relates to potential protective factors that you brought up, whether it be environmental, genetic, etc. And I want to ask if you could say a little bit more about these potential protective factors.
Yeah, I think that's an extremely important question. Let me try to be brief on this. Part of this new science is that we now have the capacity to ask questions about protective factors, which I really don't believe we had a meaningful way to do before. We've had a tremendous number of hypotheses, and obviously we can all weave yarns about what could be a good protective factor, but we haven't had scientific evidence on which to base hypotheses that we could then test. So, of course, genes identify mechanisms of risk, but every gene also has another variant in it that presumably people who are at risk are disadvantaged in not inheriting. So that other form is a potential protective form. So the other reason that studying family members like siblings is so important, because presumably they manifest two differences from their ill sibling. One is they don't inherit all the risk factors, but they also theoretically inherit some protective factors. So we can begin to explore those in healthy relatives. The other point is that one of the reasons to study genes is to finally have a way to approach the environment. Because the problem with studying the environment without understanding people's genes, it's like trying to understand how you bought railroads in the Monopoly game without knowing how much money you had. The problem is everybody negotiates the environment differently because everybody has a different toolbox, biological toolbox, with which they handle the environment. And we all know that to some people, stress is an overwhelming difficulty. To other people, it's an invigorating challenge. It's very difficult to deal with environment without knowing the individual biological factors that people negotiate their environment with. So now that we have these genetic clues and this foothold in the complex organization of brain development and function that's related to psychiatric illness, we can ask much more meaningful questions and design scientific experiments to identify protective factors. So I think we're going to be seeing much more of this emerge over the next couple of years.
What another area that this is bringing up some questions about relates to the genetics, and that there are so many different genes involved, and the fact that schizophrenia looks different in many different people. And the question is, as we get more information, will we begin to say that what we now call schizophrenia is really more than one illness, or is it one illness? And how does the genetic information inform that?
I think, and Jeff, you're very knowledgeable about this. So, I think we all know as clinical psychiatrists that our diagnoses, are not valid identifiers of biological conditions. These are descriptors that have evolved over time to help clinicians have a more structured approach to the treatment of patients and to informing families about likely outcome. But they are not, based on an understanding of how people got to the behavioral states that they're in. They're just not. And so we certainly assume that there are many different conditions grouped under these diagnostic labels. And I think we will increasingly tease them out. I mean, as everybody knows, at the turn of the 20th century, there were many conditions that were called schizophrenia, and many of the infectious diseases that affected the brain. were given the diagnosis of schizophrenia. They were slowly but surely chipped out of that otherwise, very heterogeneous population that got the diagnosis. We will probably do some more of that over the next decade or two. But clearly, there are certain ways a human brain does not develop normally. There's not an unlimited number of ways. There are certain ways, and that probably there will be a number of different causes that converge, almost like a funnel, onto this particular syndromal presentation that earn patients these diagnoses. There are many different reasons that you can be depressed than ways depression feels. Depression has a certain way it feels, ways it presents, but there are clearly many different reasons that you get to that state. And we think that's true about intellectual development. We think that's true about schizophrenia. It's certainly true about autism. These are, in many ways, developmental states. I guess one of the challenges for psychiatric thinking that's emerged from this new science that has really been led by genetic discovery is whether these are true diseases in the traditional medical sense of the word, or whether these are states of brain development that have a particular way of manifesting themselves in our complicated human environments that change as people age and develop. So that's, I think, one of the challenges that we face in getting a better foothold on understanding what our diagnoses really represent. I want to say one other thing about treatment, because I think it's very important to realize that we now have the potential, which we've never had before, to find treatments based on causes and not phenomenology. And this is the hope of all of medicine now. And this will allow us to do what is another holy grail in medicine right now, to individualize therapy. We have no approach to individualization of therapy now. It's trial and error. But by having biological information about how people are different, and I think the questioner asked the key question, people get this illness and they manifest it differently. They respond to drugs differently. Their outcomes vary. Their capacity to adapt, to make the best of it, vary enormously. This variation has something, at least in part, to do with individual environments, to socio-cultural and economic opportunity, but also to genomic differences between people. Some of the reasons people have clear differences in how they respond to drugs is their genomes handle the drugs differently. By having this kind of information, we can begin to chip away, and it's not going to be a straightforward, easy, quick process, but we will begin to be able to chip away at individual differences that we can actually control. And so we'll maybe be able to come up with a smorgasbord of treatments that won't just be random or trial and error, but will actually be informed, and we'll be able to make reasonable guesses of what are the likely best combinations, just as increasingly being done in cancer now, because we'll have some more scientific evidence to base those decisions on.
So similar, just now it is often trial and error and clinical judgment. In theory, we'll be able to do a blood test to get the information about the genetic makeup of the individual and correlate that information with what we know about the types of medicines that people with that genetic makeup are more likely to respond to. Is that the way that would work?
I think that's a very correct statement.
Okay.
That's where we're heading.
Yeah. One of the great tragedies of this illness is when a person dies from suicide. And we have a couple of questions about suicide risk in schizophrenia. And I'm wondering if there's any information in terms of some of these genetic studies and the issue of suicide.
So I think you well know that the lifetime risk of suicide in patients with schizophrenia is over 10%. I mean, it's a huge increase in the risk potential, and suicide is one of the major risks of all major serious psychiatric illness. So there's a lot of interest in understanding the determinants of suicide. And as I think many people listening to this realize that many people who commit suicide are not chronically depressed. And many people who successfully commit suicide do so with the people even close to them having no idea that they're on the verge of suicide. Many suicides are impulsive. and many suicides occur on a spur-of-the-moment impulse. So there's a lot of interest now in trying to understand what are the risk factors for that outcome. I don't think we have good data on this now, but this is an active area of research, and the research is being done in very similar ways that the research is being done for studying schizophrenia or bipolar disorder, looking at genes that are in suicide attempters and also in suicide people who actually commit suicide, because one can regrettably look at the post-mortem tissues of those people and try to understand what was the proximal act at a molecular genetic level that led to the suicide. I think in the next 5 years we will have some clues to factors that might be useful in predicting who is at increased risk of impulsive suicides. So I think this is another area where there's going to be some progress.
In terms of the issue of genetics and medication, tell us a little bit more about how the new information that we're developing will also help us develop new medications that have different mechanisms of action and that might be more effective in treating both the positive symptoms of schizophrenia and also the negative symptoms.
So I think there are two ways genetic discovery is going to help this. First of all, some of the genes themselves are targets for drugs. So this calcium channel that I mentioned is clearly what's called, the people in the pharmaceutical industry would say, is a druggable target. You can create small molecules that target that. We can't just throw drugs at it. We have to understand what that gene is telling us about what's wrong in the calcium channel protein. So that requires some basic science work, but many groups now are exploring this. Once we find out what's actually wrong with the calcium channel, we can target that abnormality using the modern technologies of drug development to try to find a drug that targets that. And actually, in the individual medicine world, individuals who have the specific genetic variation in that calcium channel would be the prime targets for any drug developed based on that calcium channel. So that's one way. And then there are other targets, proteins. Genes primarily are the blueprint for building a protein. A calcium channel is a protein, potassium channel is a protein, dopamine receptors are protein. So many GABA receptors are protein. Many of these molecules that we've been interested in are proteins that we can target with drugs. The other thing is genes are entry points into how cells do their day-to-day business. So while we can't necessarily target a lot of these proteins directly, we can target the effects of the protein. So I like to think, if you think about a cell as a domino setup with all these dominoes that are pushed down and they go along all these pathways, and that you may have a glitch, you may push a domino down at one end of this complicated array of dominoes all connected to each other. You may not be able to do anything about where that first domino goes down, but that train of falling dominoes may feed through some central node that then splays out into a whole series of other expanding falling dominoes. You may be able to target that hub. So you may be able to target some downstream effect of the glitch in the gene, even if you can't target the gene itself. So that's the other strategy for target discovery based on genetic findings. The hope is that all of these genetic discoveries will clue us in to how the cells got going on the wrong path from the very beginning. And so there's going to be, I think, a whole, sea change in strategies to find to find therapeutic molecules. And this is the same thing that's happening in cancer now. It's the same thing that's happening in all of medical science. These clues are developing new assays, new strategies for finding what went wrong in the basic programs that got the organ, in our case the brain, going to begin with. And actually, this is true in many areas of medicine, that many of the diseases of adult life are really diseases of how these organs develop. And certainly that's true in mental illness. These are disorders of how the brain develops. And these genes are about brain development. And we're going to have to explore, if we're going to look for curative therapies or maybe even preventive therapies that target the problem we have to try to explore this in new ways.
You took the words out of my mouth because I was going to bring up the question of prevention, not only finding a cure, but as importantly, ultimately, ways to prevent the illness. When you're looking down the road at where these discoveries are going to bring us, do you see that as something that will happen given the information that we're developing?
I think, you know, my expectation is, and I'm one of these guys that always sees the glass as half full. So I'm a half full glass kind of guy. You know, I think we will do this to some degree for some groups of these conditions more than other. But I see no categorical reason why we cannot reach a stage where we can have prevention strategies. I think the more we understand the absolute mechanisms of illness and how risk translates into illness, we will have approaches to do something about that inexorable march to illness from risk. People inherit risk factors and they accumulate risk factors. The manifest illness is the accretion of all these accumulated risk factors. So there should be steps along the way where intervention would prevent the dominoes from getting all the way to the end. We want to stop these falling cascades of dominoes that accumulate from getting to an end point where it's no longer possible to reach some compensation. And all I can say is the more scientific information we can accumulate that is relevant to these questions, the more we can probably come up with strategies to do exactly what you asked, Jeff. We have to invest in, and this is what NARSAD does. This is why NARSAD is so important, and the Brain Behavior Research Foundation through NARSAD grants has been so critical, because it is a recognition of how critical it is to invest in getting this information.
Well, Dan, I want to thank you, first of all, for an extraordinary presentation and discussion today, and more importantly, for the work that you've done over the years and the work that you're doing now and the team that you've put together. It truly is an area of great hope. And I feel inspired by hearing about the work that you're doing. And based upon the questions and the comments from our audience, I know that they are too. So I want to thank you, Dan, for an extraordinary job. I want to also thank our audience for joining us today. The foundation, through its research grants, is dedicated to improving the lives of people with psychiatric conditions and ultimately finding the cures and the ways of preventing these illnesses. All of the research that we fund is made possible through private donations. So anybody who's joined us today and would like to make a donation, large or small, please visit our website, bbrsfoundation.org, or call us 1-800-829-8289. The webinar has been recorded, so if you missed any portion of it or would like to share it with family or friends, you can visit the webinar page on our website. And I hope that people join us for our next webinar. It will take place on Tuesday, April 16th, 2 o'clock Eastern Time. And we will hear from Dr. Husseini Manji, who's a member of the Foundation Scientific Council and is an adjunct professor in psychiatry and behavioral sciences at Duke University School of Medicine and vice president of Janssen Pharmaceuticals. He will speak to us about bipolar disorder, harnessing brain plasticity for improved overall outcomes. This concludes today's webinar. Once again, thank you, Dan, and thank you, everybody, for joining us. Take care.
My pleasure. Thank you, everybody.
Bye.